Many of you may have heard of Dr. Neil Spector. He was a celebrated Duke oncologist, and in the prime of his life and his career, all of a sudden, he couldn't function. His heart deteriorated to the point where he needed a pacemaker installed and eventually a heart transplant. And even though he was a doctor, he couldn't get a proper diagnosis. But through persistence, he figured out the root cause of his illness. What was it? Lyme carditis. Neil wrote about his journey with Lyme disease in his memoir, Gone in a Heartbeat. He then took his research skills in cancer and geared them towards solving the problem of Lyme disease. He partnered with Dr. Timothy Haystead, and the two of them were looking at solving the problem in a completely different way. Diagnosing Lyme, treatment of Lyme, using approaches pioneered in the cancer industry to solve this infectious disease problem. Unfortunately, Neil passed away in 2020, way too soon. But his work lives on. Here we interview Dr. Haystead and learn about their groundbreaking approach, which we hope will absolutely revolutionize the way that we treat tick-borne illness. Well, Dr. Haystead, it is a pleasure to meet you and to talk about what you're doing in both Lyme diagnostics and therapeutics today. Mm-hmm. But before we start, I wanted to just say, I've worked in technology for quite a long time and working at commercializing technologies. And one of the things that I've found is a lot of the biggest inventions or the biggest breakthroughs come when you take something from an adjacent sector and then put it into the problem you're trying to solve. So I worked in medical devices, and sometimes you could take something from automotive or aerospace and then apply it and get a breakthrough. And that's definitely what I see in your work, taking some technologies from cancer and applying it to infectious disease, more specifically, Lyme disease and Bartonella and tick-borne illnesses. And so I'm really excited to talk a little bit more about your work and the science behind it. But before we go there, maybe if you could just start with a little bit of the backstory and how you got into, in the cancer arena and then got interested in Lyme disease and trying to enter that problem space.
My lab is a chemical biology lab, but it was born out of when I began my independent career as a postdoc, we were interested in protein kinases, signaling, single transduction inside cells, how cells communicate with hormone binds and it affects the cell, and then we were using inhibitors, like small molecules. So we sort of started to be very early on in my career, and that eventually evolves into a company that I started in 1990 called mid-90s, I guess. We were looking for money, and called Therapeutics, and it was based in downtown Durham, and we developed this chemical platform to target enzymes that were thought to be good drug targets, and it worked quite well. And we developed these highly molecularly targeted inhibitors, based on chemistry. So you could take an enzyme that had been identified to be involved in cancer or some sort of cancer specifically targeted. And that's what the company was about. And it developed, out of all that came an inhibitor for a protein called HSP90. Mm-hmm. And that ends up in clinical trials. The company discovers it. It ends up in clinical trials and gets acquired by Pfizer. And so it was a molecularly targeted ligand, if you like, or drug, very specific. And then, so that was very exciting. We could have done this for dozens of other types of targets at the time, but we were a company and we had to have focus. So when the company gets bought by Pfizer in 2008, I got the right, if you like, to practice the technology. It took about 2 years. Back and started to work on it in my academic lab on academic topics. And at that point, I sort of, had started to develop the idea that you could use the same molecules to image things. Mm-hmm. Like if you could attach things to them, and like fluorophores or isotopes and things, you could learn something about the biology of the disease, if you like, by imaging it in real time in animals and then ultimately humans. And that actually worked too, so it was like this progression. And we had a large grant from the DoD for breast cancer with a colleague here, Dr. Liley, and took two things into clinical development, into phase 1 here at Duke, funded by the Department of Defense, of all things. But at this point, Neil had come into the picture, Dr. Spector, and I was given a talk on using the same approach for malaria, which was another interesting. There was a really great colleague here, Dr. Emily Derbyshire, was a malaria. Anyway, we were thinking about using HSP90 inhibitors that would specifically target the pathogen form and not the human. So same chemistry that we use to molecularly target, disease proteins in humans and not the normal one, right? So you're trying to discriminate one from the other. And he said, "Oh, could you do that for Lyme disease?" So I go, I don't really know about Lyme disease. But, so he tells me, so then just Neil's story. And I actually worked with Neil during the Serenex years. So he did a deal with them when he was working for GSK. So he said that the technology platform that we developed actually worked.
Yeah.
And they got there, so he was impressed by it. So, if I said after that was, he would believe, So, and we thought, so we started looking at, became really interested in taking pathogen genomes like Borrelia, comparing them. So, with evolution, we all know about, right? So, a lot of proteins in us are conserved in organisms that go all the way back to the dawn of time, right? And so if you compare like the HSP90 from the human to the Borrelia version or any other prokaryotic version for that matter, you can see that they're related. They're called orthologs, but their amino acid sequences are different. They're subtly different, particularly in the way they bind their normal enzymatic substrates, like a thing called ATP. There are differences that can be exploited chemically, right? So the platform we developed to develop the original HSP90 inhibitor, you could very easily translate that to the pathogens. So we spent, and that's what the program became about, developing a specific inhibitor for this one target. It would not bind to the human, but it would bind to the Borrelia, and that's what we've actually done. And the Cohen Foundation funded it originally, and then Bay Area picked it up. And as of last year, we actually have a ligand now that is super specific for Borrelia.
Yeah.
And then, and so the idea with that is then to take what we did for cancer, develop this into an imaging agent, a diagnostic tool, and as a therapeutic.
So before we go there, you've got HSP90 in humans, and then you've got HTPG in Borrelia. So what do these proteins do, and what makes them such good targets for therapies?
And it turns out, and I think it's a bit of a paradigm shift in how we think about antibiotics in general. We just submitted a paper for publication on this idea. There are certain characteristics of HTPG that make it ideal for this type of approach. One is it's a very high copy protein. That means the bacteria cells make a lot of it, and so do humans. Which makes it an ideal imaging agent, because if you can get a lot of molecules into the cell, you could see it. So it's high copy. It's a non-essential gene, which is another plus. It means as an traditional antibiotic target, it would be of no use, because inhibiting its activity wouldn't do anything, and genetics have shown that. So it would have been ignored as an antibiotic target. So that's important. And its role in the organism is at every stage of its life cycle. It actually functions in protein folding and chaperoning. So when proteins are translated off the ribosome, it acts as a scaffold, if you like, to enable the protein to fold correctly. So that's what it does. So it's always there.
Okay. So
that's important, obviously. So and it ticks all these boxes. You suddenly realize that it's becoming a model for other things too. So
HSP90 and the HTPG. So they're, the interesting thing is these proteins are conserved, right, across bacteria, viruses, human cells, but they're also, as you said, kind of, they have different amino acid sequences in that binding site. And so one question I have is how specific are they? So you've been working on making sure that it doesn't bind to the human tissue the same that you're binding to the bacteria, but what about other bacteria? What about other spirochetes? What about
very good question.
Other is it specific to the genus or the species of Borrelia? Like, what do you know about that?
Yeah, we started to really dig into that, and actually we have at the moment we're repanning our library against Dentacola and the Palladium, which is a syphilis version, and lepto or something. Another one, and even Babesia, which is a bit like malaria, right? So,
right.
They all have their HTPGs, and interestingly, if you compare, like E. coli is a good one, like it's a common gut one, to Borrelia, there's about 80% difference in the amino acid sequence in those sites, which is actually the same 80% difference between us and human and them. And it's like they are as different from each other as they are from us.
Yeah.
So it becomes almost an endless game you could get into. Like species-specific ones for, Dentacola, say, if you had a reason to do that, might solve dental caries. Syphilis is actually a problem now. We've got resistant species emerging, so you can imagine it working for that. Or you could have one that's a pan. All these, I think some of the one, whenever you begin this chemistry effort, it's an iterative process. So along the way you generate all these molecules, some of which are going in the right direction and some that aren't. But you repan them against these other species because there may be a probability you unwittingly made an awesome inhibitor of the Dentacola form, So, so that's the beauty of it all. And the chemists in my lab are extremely talented. We're creating this enriched chemical library that can be then panned against all these other species as the starting places if you want to do that.
Yeah. Well, and this is critical because, you need to, if you're lighting something up in an imaging aid, you want to make sure you're lighting up the right bacteria and that you're not lighting up E. coli or even another spirochete like syphilis. And so, so it sounds like what you're saying is that you can tune the molecule and then look at the binding and really choose the ones that can be specific. So, but describe that process a little bit. You've got the HTPG and you've got a molecule that binds to it. And then you can tether that to something that allows you to either diagnose or treat. Maybe explain a little bit about that tethering process and then, how you use that just start with imaging first and then we'll move to therapeutics.
The first thing, the easiest thing to do in a lab like this is just to generate a fluorophore version. So you have the ligand, and my arm's that, and then you got me would be the fluor, right? So,
something that basically fluoresces that you can pick up on a microscope.
And there are hundreds of those.
Right.
And that's important. There are hundreds because they do affect the pharmacology of how these things, when you inject them, some are very greasy, some are very polar and water soluble. So all that matters. So that's an easy bit. You can change that, and then they all different wavelengths. So we have red ones that have low background. We like to do those. And so once you've got those, and that gives you a way to now access the sort of biology of the target in real time. So we know we can add these probes to Borrelia, and they're still alive because it's a non-essential gene. So you can see them wiggling around with the probe in them. So that's the start. So you know the ligands work here. You can compete the signal and all that stuff. And then you start to think about, well, this isn't necessarily useful for whole body imaging, right? It might be useful in histology. And we're doing some work with Amir Honzik where we're taking this fluor probe and he's injecting it into the mice, and then he's going to take the joints and slice them up, and then do histology and say, can we see these 10,000 per milligram beasts? Where are they? In a whole mouse even. So that counted up to humans. Really, that's not very useful. Maybe you could use the fluoral version of the blood test that we discussed already. We're looking somebody working on that to see. There's some literature. It's one of those. Is that real? Does that really happen to people? Don't know. But we're looking. We certainly do know it binds to platelets. We'd agree with that literature in culture. We could enrich platelets from humans and get a better chance of finding that 1-2 areas. per milliliter, whatever it is, that would have to be there, but if that's real. But PET is the one that comes to mind as the ultimate method. So we're now working on chelates. We have a couple of ideas there. There's quite a dynamic range of different types of PET agents you can use. We're working with the Department of Energy to get access to some of their latest ones.
So basically now you're working with these different PET agents, trying to test them to see what goes where, essentially. And then the idea or the end game would be that you could take it, inject it into somebody or an animal that's infected, and then image it in a PET scanner so that you could see exactly where it is.
Yes.
And so do you think that that's something, do you think that the PET agents exist where that would be like a local scan? Do you think you could do it systemic? Whole body. And what about the blood-brain barrier? Are there PET agents that can actually penetrate?
Well, that's, again, that's another iterative process. So I don't know. All I can tell you is that the brain group got interested in the, I got interested, actually, wasn't they? We're interested, I got to them. The glioblastoma is a big issue, obviously, and especially some very aggressive forms in children. So they have the same issue, getting across the blood-brain barrier. And with them, it might be an easier bar because when a tumor gets in the brain, it actually disrupts the blood-brain barrier. No matter what people say, I mean, it's gone.
Well, but it could be the same for infection too.
It could be exactly.
Yes.
I think it's such. What is the blood-brain barrier? It's actually in the vasculature. It's not this thing at the neck or something. It's actually the whole vasculature. There's very little known about it, really. Actually, there's people that study it that much. They just know that it won't let things in, which is probably a good thing because you can imagine if some of the drugs we take could get in easily, we would want them in there.
Unfortunately, Borrelia is sneaky and it can get in.
Yes, exactly. So my feeling is that that's going to disrupt it's going to be in that interstitial space. It's not generally thought that it penetrates cells.
Yeah.
We haven't been able to do that. We've certainly shown that it sticks to platelets in really disturbing ways. But I don't think it gets in. So it's going to be in that interstitial space that, may well have access.
So when do you think you'll be at the point where you can start testing these imaging agents in animals just to see where it goes? Now.
Actually, tomorrow I'm having a conversation with Bay Area, not to put them on the line, but I'd asked them a year ago. There's a guy called Matt Krogan I've been working with that Neil introduced me to, and we decided that we just get a PET machine, small animal PET machine, and try to find somewhere where that's got a small animal PET machine, and they're doing an infection model. And it does not exist.
Right.
So we're going to have to do it here. We suddenly realized, and that is the only obstacle in the way of going into clinical trial. It's what we've got to show the FDA.
Yes. I hope you can get that barrier overcome because the ability to image Borrelia and see where it is in the body would be game changing.
Right,
really.
exactly. And human might be the model, you see. So now so let's take a COVID, right? We jumped all of the safety studies into human. The advantage of imaging agents is that they fall under microdosing, which is a lower safety barrier than a drug.
Yeah, because you're not giving it for long periods or large doses.
Exactly. Correct. So we may be able to play that with FDA. These are things that we've got to go and try. You know? And I think we can, we can make, if we don't have out there, we have safety. We can do safety. Is it what, and then you've got the hypothesis that it's going to target this organism, but the model is not good enough to really, and often models don't translate to human anyway, so it'd be an interest. That's why I needed Neil, you see, that's the thing, you needed somebody that could have made that argument, that there's an unmet need, and it's a problem. But the climate has changed. I mean, it'd be interesting to do that, and to try and say, we've got GMP material, GLP, safety. We believe that, if you give this to a person, it won't kill them and it won't do any harm, more importantly as well. And we can follow this radio tracer that's used routinely in medical. That's why I keep assessing about using things that are in current medical practice.
Yeah.
So I think you've taken a number of boxes, and we've got a doc willing to do this. We've got patients that are willing to do it. And we don't think we've hurt them in any way. And then there's even, there's the cool thing is there's even a clinical trial going on at the Cohen Foundation of funding, for newly infected patients with Lyme. These are like the ideal model, right?
Right.
So,
right.
They've got the disease at the bullseye. Could we slip them some of this stuff and
then image it and see where it is?
And see where it is.
Yeah,
and then
absolutely.
You got it, right?
Right.
And you got it.
Right.
So now we can talk about other things. I think that's the path, and we're gonna, need help with that.
Yeah.
So.
Well, and that's extremely exciting because, like I said, being able to image, it can go to so many different places in the body, and so knowing where it is will help inform treatment. But the technology and the binding can also be used for therapeutics, right?
Yeah.
Which is something completely different from the approach we take with antibiotics, which is not specific and has challenges and potentially not effective.
Yeah.
So maybe talk about how you can take that same molecule and then now make it a therapeutic instead of a diagnostic.
Yeah. We call those berserkers.
I love that name.
Right, because you're trying to sneak this thing in that's been seemingly benign,
right?
And then you activate it, and then it causes havoc intracellularly. And we have a couple of those. One is also the PET agent. You can put things called positron emitters, which are how we see PET, positron emission spectrum, tomography. But there are other ones that emit things called alpha particles, which are things like that are they're nuclei that are traveling and they smash into DNA and they smash into proteins. But they can only do that within the cell internally, and they cause just, for a treatment of thyroid cancer, that's what they do. It's one called 131. It's been used for 50 plus years to treat thyroid cancer absolutely effectively. Well understood. We could put that on one of these things. That would be our iodine carrying probe. And then there's new isotopes coming out, with a few around anyway, that emit alphas, one called actinium-225. That's a new one from the DoD, which can also be seen by PET, so you can see both. And it would deliver an alpha particle. Again, microdose. That's the other thing that we really, when you normally use these things in medically, because they're often not targeted. They either put something, like you're finding a nuclear weapon in a little particle in you.
Yep.
But we would have the opportunity to exploit the pharmacodynamics of these things, where you would have a microdose that you could give systemically that would find the bug and get concentrated in it. So it would be at the level that it would be as if you put it, if you could see single molecules and you could inject them. Yeah. It's just using uptake. You can measure that by PET. So I think that the therapeutic window that one can achieve with these things could be enormous, because you can get it to concentrate. That's why the copy numbers are important. Get enough in there.
Right.
And the affinity is high enough to find it, all that sort of thing. That's the beauty of PET, you see, because you can see it in real time.
Well, that's the beauty
of it's real. You can see it.
Yeah, that's the beauty of your approach, is that you can image, see where it is, then you can use the same binding mechanisms to basically go in and instead of now tethering an imaging agent, you're tethering some sort of toxin or that's going to kill off, and then you can go back and image and see if you've actually killed them all. Did everything. And so I think that iterative loop is what's been missing in the Lyme community. There's people go through treatment and they don't know whether they're well and whether if infection is, they know whether they're not well, but they don't know whether the infection is what's causing it.
I mean, from a business point of view, I see it's quite an expensive thing. So you can see people could make money on it. Let's be crap about it, but that's the driver for when you get investors involved, isn't it? People would hate it, but it's
effective. And I think when you're keeping people from basically a life of chronic illness, right? That's critical.
And that's what it's for. It's not to treat. Doxy works for if you get it early enough. Ampicillin will if you get it early enough. so somebody in this situation where they've got this underlying infection, and if it is because there's a bacterial infection there, we some numbers that we can come where we could see it.
Yeah, they've been through treatment, they're still not well.
Yeah.
Is it really caused by Borrelia or something else?
Yeah.
And then how do we treat, and then is it, do we get rid of it essentially after that treatment?
Maybe it's you end up saying, well, this is really an autoimmune disease, and immunomodulatory therapies are unique,
Right.
Which is another forefront in medicine. It's really got some game changers in there too. That really work. They're not, unfortunately, not cheap. So those drugs either. So, but they are there. But you don't even want to use them. If it was that, not something else, it wouldn't be much point in using them if there's an underlying infection that's just, you've got to immunosuppress somebody.
Right.
Not a good, So again, that's the importance of the whole thing, and why we're doing it really is to try and discriminate those things.
No, and I think that's a great point. This isn't meant to be a frontline therapy. It's something that would be kind of for people who are chronically ill and having challenges with treatment, and really have run out of options. So you said in the diagnostic space, you have, you're right on the precipice of kind of going into animal models. Where are you in terms of the treatment paradigm?
Same thing, you see. It's the same. We would do the same thing. In an animal model, you would not only image it, then you start giving it alpha emitters. We have these other ones that are chemicals. There's one called vertephorin that's been used for macular degeneration for years. When you excite it with light, it generates what's called nature's bleach, which is reactive oxygen.
Yep.
And they're kind of interesting because they'll trigger programmed cell death rather than blow the thing up. That's important because it can improve immune surveillance, and things like that. So
because that targets your immune cells to come in and help with the process.
So we've got one of those. So the whole thing, what's really exciting about it is it changed the paradigm about how we think about antibiotics now and what are our targets.
Yep.
So Bartonella is the other program that the Cohen Foundation. So we're looking to see, can we do the same thing with Bartonella? Develop a diagnostic and a treatment. And we have 2 targets that are going really well. We've got 3 co-crystal structures on 3 different molecules targeting a protein called GLIN2, glutamate synthetase. It's an abundant, non-essential gene.
Yeah, so it's the same approach, just a different molecule.
Yeah. So then you think, so we found a whole class of enzymes. They're called tRNA ligases. That are the enzymes that take the tRNA with an amino acid on it, and they take it to the ribosome and they transfer that amino acid to a growing polypeptide chain. So they're abundant too. But what's really weird about these enzymes is that every single bacterial species has a completely different sequence for those tRNA ligases. There's 20 of them.
Wow.
So those could be a great diagnostic target. Even Borrelia, we've got one for Borrelia, we've got one for Bartonella. If you align them, you do they don't even look like they're related. For some reason, they've conserved their active site. But there's tremendous diversity in the binding. It's like this goes against the grain of evolution.
But that's a fascinating thing to exploit for specificity, to make sure that you actually have what you're targeting. That's great.
So we started to fuss with that one now, and they actually work very well with our platform. They look like things called protein kinases. They bind these their ATP, like these enzymes that's a strength for us, like our HTPG. It's in a specific orientation, and it's highly amenable to manipulation through chemical biology. And those could be blood tests for all kinds of pathogens. They could be ways to deliver. So the way that I often talk about now with the Borrelia, it's a model. It's such a great model that could be if it could work here. Well, we've got this massive problem with dealing with infectious disease in general. We have antibiotic resistance everywhere. Borrelia co-infections. We know they immunosuppress, and we know that other organisms do the same. So it's a real problem. Yeah. So what if we start targeting housekeepers?
No, I think it's a fascinating approach because these are, Bartonella and Borrelia are 2 organisms that they're more in your tissues than they are in your blood, and that makes them very difficult to find and diagnose. And so I think your approach is, as I said, really novel and fascinating because
Yeah.
it's different and has a lot of potential. So
Yeah.
I really appreciate you taking the
Yeah.
time to speak with me today and
Sure.
to tell us a little bit
Yeah.
more about it.
Yeah.
And I hope you get that PET scan or the PET machine funded so that we
I-
can keep that
I do have it.
keep going.
Yeah.
So that would be fascinating.
Yeah.
I look forward to seeing all
Yeah.
the great things ahead.
Yeah. Thank you.